Hello and welcome dear students, pharma professionals and learners to the very interesting topic on impurity qualification. The current topic is for impurity introduction, the types of impurity in pharmaceutical formulations and APIs, and what are the ways to qualify the impurities if the impurities are above the qualification thresholds. So in this video you will be walk through the impurities topic, and mainly the impurity topic will cover the NGIs, means non-genotoxic impurities. The other impurities will not be discussed in the present video. The separate video will be coming on the other type of impurities. So in introduction we will see what is impurity. See, impurity is any component of the new drug substance or new drug formulation or new drug product that is not the chemical entity defined as the new drug substance or the part of drug substance in the formulation. If you see the formulation, you check the impurity profile. You have the impurity profile in your hand, and that is prepared or that is taken based on the impurity method development, that is RS development. Impurity is also called as related substances in the general terminologies. Impurity types are there. Mainly we can divide the impurities as organic impurities and inorganic impurities. Then residual solvents are also the impurities. But always remember, the qualification is done only for the organic impurities. Inorganic impurities and residual solvent, I didn't got any information on the qualification. That's why the elemental impurities and residual solvents are required to be limited and their presence cannot be qualified. So organic impurities may be identified or unidentified. These may be volatile or non-volatile. So from where these impurities come into the drug product or drug substance? From process it may come. It may be produced by degradation. Sometimes the API has enantiomeric impurities. Then out of these, two main types of impurities are genotoxic impurities and nitrosamine impurities, and these are highly toxic. Now the genotoxic impurities and nitrosamine impurities will not be a topic of today's video. We will discuss only on the non-genotoxic impurities. Process impurities comes from the starting materials, maybe byproducts, intermediates, if they are not taken out by the process, then they remain in the product or drug substance and they called as process impurities. Then degradation impurities, these are drug related impurities. These may be formed due to storage conditions, due to aging, or these are the degradation products. Sometimes the drug may degrade and those degradation uh, side products are called as degradation impurities. Then enantiomeric impurities, as the name suggests, the enantiomeric impurities are the mirror images, like enantiomers we have studied. So these are the enantiomeric impurities. Then genotoxic impurities, which are mutagenic in nature or carcinogenic in nature, and nitrosamine impurities. Nitrosamine impurities and genotoxic impurities are highly toxic in nature. So the limits for genotoxic impurities and nitrosamine impurities is a separate topic, and the specific understanding is required for genotoxic impurities and nitrosamine impurities specification. Then coming to inorganic impurities, mainly elemental impurities are there, which are called as inorganic impurities, like reagents, ligands and catalysts, heavy metals, inorganic salts, other materials like filtrates and charcoal. From these the elemental impurities comes into the product, and these elemental impurities are not formed into the product. These comes from the process or from the raw materials, and these are required to be limited. Then residual solvents. Residual solvents may be used during the drug uh, substance manufacturing and also in some of the drug products manufacturing. So these are required to be limited. Their classes are class 1, 2, 3 and 4. Then organic impurities. These may be specified or unspecified. Then these may be identified and unidentified. So let's see the basic differences between these terminologies. Specified means listed with the acceptance criteria. See, specified means you have given the acceptance criteria and these impurities are listed. Listed means you have listed or you have made a part of list of impurity profile for these impurities. That's why these are called as specified impurities. Then specified impurities may be identified or unidentified. So if the impurity is specified and identified, these are also known as known with the name, or you may sometime name it as impurity A, B, C, D and other names are given. Then specified unidentified impurities. These are unknown impurities with a specific RRT, that is relative retention time. That's why you are making a unknown impurity known or somewhat specified with the RRT. That's why these are called as specified unidentified impurities. Then unidentified impurities. These are the unknown impurities. You don't know their structure. You don't know their RRT. These are unknown. Unspecified, unidentified. If you specify the unidentified, it will go to specified unidentified. If you make known, then it will go to specified identified. Right. So the unknown impurities are sometimes also called as and referred as maximum unknown. So limit is given as maximum unknown impurity, and the threshold for this maximum unknown becomes the identification threshold. If this unknown impurity crosses the identification threshold, then the impurity is required to be made known. And the chemical structure and name is there, then it will have a qualification threshold. But if the qualification threshold is over for any impurity, then there will be a qualification process. If any of the impurity crosses the qualification threshold, then that impurity is required to be qualified. Higher threshold may be applied if scientifically justified, qualified or provided in the monograph. See, now you can have a question, or you may have a question that when I can have a limit more than this qualification threshold. So if you qualify the impurity and if the impurity is present in the monograph, then the qualification threshold can be widened. Lower threshold if impurity is toxic in nature. It is not always the case that you will get the ICH limit for qualification threshold. Sometimes if the impurity is toxic in nature, that time lower than the qualification threshold limits are given to that impurity. Here the table describes the limits of reporting, identification and qualification threshold for impurities based on the maximum daily dose. Maximum daily dose is the amount of drug substance administered per day. Then as per this guideline, higher reporting threshold should be scientifically justified. If you have a reporting threshold higher than the ICH limits, then scientific justification is required. And whatever the justification is provided, that should be scientifically true. Then lower threshold can be appropriate if the impurity is unusually toxic. Like the toxic word is coming many times, so you can relate this to the nitrosamine and genotoxic impurities for your easy understanding, because the genotoxic impurities and the nitrosamine impurities have a very less threshold for the drug products, because these are very toxic in nature, and these are also known as cohort of concern. That means these are mutagenic in nature and may cause or may lead to potential cancers. So potential risk of cancer is there. That's why they are toxic in nature. Now, for a 2 gram per day dose, the limits are given, and above 2 grams per day, the limits are given. This is for drug substance. Now we will go to drug product. Here, the maximum daily dose is given, and based on this, the reporting threshold, identification threshold, and qualification threshold are given. So, like the justification and all this required for drug substance, similarly, the justification is required for drug product. Now, if your product crosses the limit of qualification, that means the impurity is required to be qualified. Like less than 10 mg dose, 10 to 100 mg dose, above 100 mg dose and below 2 gram, and above 2 gram. That time, if your impurity crosses these limits, and that time you have to qualify the impurity. Then what is the meaning of qualification? We should understand this in very detail. Qualification is the process of acquiring and evaluating data that establishes the biological safety of an individual impurity or a given impurity profile at the level or levels specified. So what does it means? See, the reference product or innovator product is always qualified based on the clinical studies. So along with that product, the impurities are also get qualified because the clinical safety is proven. Now, if the product crosses those limits, like in RLD or the monograph limits or the ICH limits, that time the qualification is required. Why? Because the product contains impurity level higher than the ICH thresholds or the monograph thresholds. That time the applicant has to prove the biological safety of the individual impurity. For example, if the impurity A is above the qualification threshold, so the impurity A should be evaluated for biological safety. And if other impurities are also more, so individually these impurities are required to be tested for biological safety. Then for qualification process, the patient population and the duration of use is considered, because the drug product is being administered to the patients, so patient population is very important, and the duration of use. Sometimes the drug product is given for acute treatment, or sometimes it is given for the chronic treatment, chronic diseases treatment. So based on the duration of administration, the impurities should be qualified. Like the impurity classes we have seen, so for that the qualification tests are provided in the literature, in the ICH guidelines, and in the guideline of respective regulatory authority. Genotoxicity studies are done by point mutation and chromosomal aberration. Then general toxicity studies are done by some of the in vitro testing, and also if required, then usually these are done on the rodents, and the duration is around 14 to 90 days. Then other specific toxicity endpoints as appropriate. This is as per the product requirement, other specific toxicity endpoints and as appropriate. So these are product specific. In this presentation, we are talking about the general toxicity studies. And the general toxicity studies are based on the different approaches. These are also called as in silico tools or in vitro methods. So first is TTC level application. And NGI means non-genotoxic impurity. First of all, if you should apply the TTC approach for the impurity. If it is below the TTC level, then there is no need to qualify it, because you have applied the TTC approach of genotoxic impurity to your NGI impurities. So this TTC is threshold for toxicological concern, and the limit is 1.5 microgram per day. If it is well below that limit, then no need to go for qualification. If NGI is below TTC level as per the route of administration and systemic exposure, and it has been confirmed for no pharmacological activity, then no further study. Then another approach is structure activity relationship. It is popularly known as QSAR approaches to evaluate pharmacological and toxicological activities of the NGI. This toxicological evaluation depends on the route of administration, intended use, availability of comparable compound data. Then NGI may or may not resemble to the API structure. NGI bearing the structure not related to the API, RAX approaches may provide relevant safety information. This RAX means read across approaches. So the meaning of this terminology is that the qualification is based on the route of administration, intended use, in which condition it is being used, that means the product is being used, and is there any available data for comparison purpose for the NGI activity. NGI may or may not resemble to API structure. Sometimes if the impurity is relevant in structure to the API, these are called as related substances. Then if the NGI is resembling to the API structure, that means it will have a less toxic impact. And if it is not related to the API structure, then you can go for QSAR approaches by read across approach. That is known as RAX approach. And this RAX approach may provide a safety information. So see, these databases have the information on the impurities, their structure, and the impurity toxic nature. So these databases can be used as a starting point for QSA or RX approaches, and also these databases can provide the very good information on the TTC values if the TTC values are available. Examples are IMI and ETOX databases. The use of databases will lower the failure rates and also lower the cost. It also reduces the animal testing. That's why the databases are very important. Then toxicological databases, quantitative approaches. These are called as QSAR approaches. These evaluate pharmacological and toxicological activities of non-genotoxic impurity. Structure activities relationship approaches are also used for getting the useful information on the nature of impurities. Read across approaches are used very popularly for many of the impurities, and these are based on the QSAR principle. If NGI shows low risk, then no further study is required. See here, if your impurity is higher than the qualification threshold, and from these approaches you get that the NGI is having low risk, then you can stop the study there itself and you can move on. If the approaches described in previous slides is not working, like QSAR approach, then the database approach and TTC level, then comes into the picture the actual study of the impurities in the rodents and non-rodents. So here in this the actual animal testing is done. General toxicity studies involve the use of one species, usually 14 to 90 days, and this is to be judiciously justified based on the intended population, patient population, that is patient population, and the duration of use. So it involves repeated dose toxicity. In this, rodents like mice and rats are used, and non-rodents like dogs and mini pigs are used. These are used to detect the target organ toxicity also. Like at a relevant exposure, toxicokinetic studies can be done. Histopathological screening of fertility, this study can be done. Local tolerance might be included. Then duration of toxicity study is dependent on the intended duration of the treatment. So the intended duration of treatment is very important. Also the dose and the patient population is important while providing justification for impurity qualification based on the general toxicity studies. Then general toxicity studies, one species usually 14 to 90 days is done. Single dose studies may be appropriate for single dose drugs. See, if the drug dose is single, that time single dose studies may be appropriate, and in general a minimum duration of 14 days and a maximum duration of 90 days would be considered appropriate. In this type of approach, the 3 Rs principle is must. This principle is must for understanding. It involves replacement, reduction, and refinement. These are the 3 principles for animal use in impurity qualification. So, if possible, replace the animal requirement. If possible, reduce the animal requirement, and if possible, refine it. That means judiciously justify the actual requirement. So this 3R principle is very important. The API dose and duration of treatment is very crucial in impurity qualification, and it is case by case basis. So all the impurities are different. Every impurity have its specific structure and the specific toxicity. So the duration of treatment and the dose is very important while going for the general toxicity studies. For a drug with low dose, these studies become very difficult. So there are some approaches for qualifying, and once you qualify the impurity, that means if you found the impurity is non-toxic in nature, then you can increase the level. That is the level above the qualification threshold or above the monograph level. But if it is found toxic, then that impurity should be limited, and there will not be any relaxation for the specification of that toxic impurity. So generally in pharmaceutical industries, the impurity profiling is very crucial, and all the measures should be taken to reduce the impurity levels, or the measures are taken to decrease the degradation, like that may be storage condition or packaging or any process or drug substance purification process. There are number of processes through which the impurities can be decreased or can be kept below the ICS thresholds. Because qualification is very difficult process, it requires time and costing. So in this video, we have seen the introduction for the impurities, then impurity qualification for non-toxic impurities. The upcoming videos will be there for genotoxic impurities and nitrosamine impurities. So stay tuned to this channel, and also understand that the impurity qualification is a very frequently asked question in the interviews. Do like, share and subscribe to this channel. Thanks for watching the video.